Welcome to the 16th lecture of optical sensors course. In the last turn we discussed LSPR based sensors, and then we studied plasmon enhanced spectroscopies. In particular, we saw what is surface enhanced Raman spectroscopy and how we use it for sensing. Today we are going to talk about multiple optical sensors of different mechanism. For example, it will be like fluorescence, enhanced fluorescence, enhanced IR absorption, then coupling between localized and propagating plasmons, and then colorimetric and Kars based sensors. But before we go there, I want to tell you something, that we were studying metallic nanospheres, and we saw that when we have a nanosphere, then the electromagnetic field gets enhanced in its vicinity. But if you bring two of them and they are very close to each other, then the field enhancement is not large at the nanoparticles, but in the middle, you see here. So, that happens because of the superposition of these two electromagnetic fields, and here you can see that if we increase the gap on the x axis, you can see the enhancement factor goes down. So, it is like the coupling between these two breaks. So, when you are preparing a film of nanostructures, you have to consider that there should be an optimum gap to have maximum performance. So, let us take an example of porous films of nanorods. What I mean by porous film is like the nanorods are separated by certain distance, and if this gap percentage is, say, you can write it 0%, 10%, 20% or 30% like that. Here I am showing you the search spectra of 4 amino thiophenol that are adsorbed over this kind of nanorods of silver. And you can see that with an increase in porosity, the enhancement first increases. So, you can see that the green curve, which is where it comes to the maximum, and then it again starts decreasing after further increase. So, what it means? It means that there is an optimum condition when you will have maximum plasmonic enhancement from a nanostructured film, and that comes when the electromagnetic field enhancement here and here is contributing in such a way that the spectroscopic signal is maximum at that point. So, for example, in this particular case it is about 30%. Okay. So, when you make a LSPR based sensor and you have a nanostructured film, you have to consider these things into account for optimization of the structure. Let us say what is fluorescence. So, fluorescence was named by George Gabriel Stokes while he saw mineral fluoride which lights up when illuminated with ultraviolet. So, that is how he named it fluorescence. So, what happens actually is that we can understand from Jablonski diagram, that if you have a molecule, you shine light of certain wavelengths in nu A, it gets absorbed there, and after certain time it comes back to the ground state. So, that is called fluorescence. It is also possible that it may go to from, say, singlet state to triplet state, and then you will have something called phosphorescence. So, here are certain characteristic times, and you can see that the fluorescence lifetime is about 10 to power minus 10 to 10 to power minus 7 seconds, while for phosphorescence it can be up to 1 second also. So, you might have seen these kind of materials when you bring them into dark room, after certain time also they keep on glowing. So, that is phosphorescence. So, what happens in fluorescence? For example, here fluorescein, I have taken this molecule, you shine it with certain light. So, here is the absorption curve, it gets absorbed there. After this kind of vibrational relaxation, it comes down, emitting light of wavelength which is larger than the absorption wavelength. This is called fluorescence. So, this is the absorption plot, this is the fluorescence plot. If you take another example, let us say we have BOJ7 molecule or say rhodamine. So, the absorption is around 530 nanometers, that is the peak. You excite at this wavelength, in the emission you see fluorescence colors at 560 nanometers. So, this small shift is called Stokes shift. There are various proteins and compounds, say for example, you have ATGC in DNA, and then you have tryptophan and all these molecules which are present in your body even also, and various proteins, they also have certain fluorescence. So, it can be absorbed at this wavelength and they can emit. So, it is like that. I mean, without using anything, if you have a tryptophan based compound, you can measure its fluorescence directly rather putting there any fluorescent tag. So, they themselves work as fluorescence. If you want to use them for sensing, for example, what we can do is that we can have a quenching or enhancement based effect. So, for example, in this particular case you had this curve, and after you put some molecule, the fluorescence got quenched. So, by measuring how much change in intensity occurred, you can say that how much change in concentration occurred there. It can be vice versa also. So, suppose in the first case you have this fluorescence curve, you add a certain analyte molecule, you see an increase in the intensity, and this increase in the intensity is related to how much change in the concentration of the fluorescence molecule occurred. So, you can plot it like this with concentration and ratio of initial fluorescence to the final fluorescence. So, for example, in quenching you will have this straight line. If it was enhancement, you will get a curve like this. In pH sensor, you can see that the fluorescence changes with pH like this. So, suppose in this range, if you change the pH slightly, you can measure how much change in fluorescence occurred. So, by measuring the change in fluorescence intensity, you can say how much change in pH occurred. So, this can be used for pH based sensing. If you want to design a fluorescence biosensor, what you do is that you attach this fluorophore to the receptor via some linker. So, for example, I have shown here a genetically engineered bacteria that glow when they interact with the target. So, this linker provides the means for triggering a change in the fluorescence of the attached fluorophore. So, what happens actually that when you bring an analyte near this receptor, it leads to change in the fluorescence of this. So, that is why it is called label based sensing, labeled sensing, because you have to have a fluorescent tag whose intensity changes when there is a bio sensing interaction. If you bring this kind of fluorophore near a metallic nanostructured surface, plasmonic surface, then its fluorescence can be enhanced, and that is why it is called surface enhanced fluorescence. So, for example, in this case you have this copper nanostructures from this paper, and if you bring a fluorophore near in the hot spots, that is what I was mentioning, that the gap between these two particles. So, you can have enhanced fluorescence. It is also possible to have something called intrinsic fluorescence. What it means? It means that you have silver structure, on top of silver you do controlled oxidation. So, it becomes Ag2O, and then you have a bit of carbon also from the atmosphere, and this is fluorescent. So, this embedded silver rod, this works as a plasmonic structure, and in this plasmonic structure, because of the plasmons, you have enhanced fluorescence from it itself. I mean, you do not need to have any label. So, this is label free. So, you can see the fluorescence how beautiful it is, and it can be used for detection of various bioanalytes. For example, here I show you detection of glycated hemoglobin, which is found abundantly actually in diabetic patients when glucose goes and binds on the hemoglobin. So, if you have lots of glucose in your blood, it goes and binds on hemoglobin. So, this is also a very reliable test rather than measuring the glucose for diabetes patients. So, because it is constant over a period of 3 months. So, here you see that when you increase the hemoglobin concentration, you see this kind of numbers, because this has been translated into the physiological range of glycated hemoglobin in human blood. So, it is like 4 to 8% of hemoglobin So, that counts to this much concentration. What I am saying is that when you increase the concentration, what you see is that the fluorescence intensity goes down. So, it is like fluorescence quenching and it is very monotonous behavior. So, you can see that when you plot with concentration, you will have the fluorescence intensity showing a straight line. So, by this way you can use it for sensing applications. Surface enhanced IR absorption. It also works on the same principle, worked for surface enhanced Raman spectroscopy and surface enhanced fluorescence. So, you have this near field enhancement in the vicinity of plasmonic antenna. If you bring a molecule here, it will experience enhanced electromagnetic field. So, its absorption cross section gets increased. It is not absorption cross section, but since it is experiencing enhanced electric field, it has enhanced absorption. So, nano antenna has to be micrometric length, because you know the plasmon resonances. So, you have to have say lambda by 2 kind of structure to excite in IR range. It can also work for visible range also, but IR we are more interested, because I was discussing yesterday that IR spectroscopy and Raman spectroscopy, they are complementing each other. So, it is very important to study IR, but these signals are also little weaker and you want to enhance them, you can use plasmonic nanostructures in the vicinity. So, that is what we are doing. What you do is that you couple the molecular vibrations and surface plasmons in surface enhanced IR absorption. So, if you have the surface plasmon curve like this for nano antennae and it matches to the IR band of the molecule, basically you can enhance the absorption of this particular band, not the ones which are falling here. Okay. So, that is why you choose a structure which has a little broader plasmonic spectrum. If you want to use it for sensing, I have shown here couple of examples. Suppose if this is the rod and if you want to have a parallel excitations like this, here you do not see it, but if you have perpendicular excitations like this, then you have lambda by 2. So, it is like antenna. When you have antenna like structure, you can see that this black curve shows enhanced relative transmittance. So, it means like absorption is high in this particular range, and enhancement factor was about 3 lakhs. There is another example where what the people did is that they started adding silver, and when they started adding silver to their study, what they found is that the absorption kept on increasing, and that is because of plasmons. So, it is the same basic principle that you have plasmons. So, you can have enhanced electromagnetic fields which enhance in turn the absorption. Also you see that they kept on increasing. Why? Because when you have nanostructure which is resonating in the IR, you have better enhancement, optimum enhancement in that region. That is why it keeps on increasing even when you are increasing the silver thickness. ESP-LSP coupling based enhancement. Till now we studied that if you have surface plasmons, you can enhance the electromagnetic field on the surface up to order of 10 to power 2. If you have localized surface plasmon, so it was like ESPs. If you have LSPs, it had about 10 to power 3 kind of field enhancement in its vicinity. Now, if you want to couple them together so that this multiplies, the idea is that to enhance the electromagnetic enhancement further, how we do is that we do a coupling between extended plasmons. So, suppose in this kind of structure, these are extended plasmons coupled to localized plasmons. You can see that if you are slightly displaced, say about 1 or 2 nanometers, you have very high enhanced electromagnetic field here. That is why it is called hot spot, plasmonic hot spot, plasmonic hot spot. And if you bring a molecule here, basically you can enhance its electromagnetic signal. So, you place this molecule here and it experiences an electromagnetic field of say about let's see 10 to power 5 in the hot spot, average enhancement is around 10 to power 2, which is similar to what you have in plasmon. So, it is like this kind of structure. You have a film, you have a metallic film here, and on top of it you have nanoparticles, something like this. They are having molecules here all here, and the spacing between them is g. So, it also varies as a function of g. So, there is an optimum condition when you have this maximum enhancement, ultra high enhancement. So, you see here for all these values of g that in this particular case that it comes around 10 to power 5 times. So, if you put a molecule here which is fluorescent, you can have enhanced fluorescence intensity, and if you go off resonance of ESP, you do not see any enhancement here. So, that fluorescent intensity goes down. So, you have a control on enhancement factor, and also you can control when to fluoresce and not. So, you can control by controlling the ESP resonance angle. Okay. Colorimetric sensors. In this kind of sensors, it is the change of color when the interaction occurs. If you remember in the very beginning of this course, when I was discussing what is a sensor, I showed an example of pregnancy kit, where you have two lines which tell you that okay, you are pregnant or not. So, it is the same kind of sensor, where just by seeing using eye, you can say that okay, this is the particular analyte that is making the change here. For example, the pH strip you take, you dip into the solution, it changes the color, and then you know how much pH it occurs. So, mostly it is qualitative, it says okay, there is a change, but sometimes it can be quantitative also, not always. Sometimes, for example, this pH strip, it can tell you okay, the pH is in 5, 6 or 7, but it cannot tell you it is if it is 5.2 or 5.5. It is also a problem with the range also. For certain purposes it is good, it is still good enough to have this kind of sensor, because it is quick and qualitative. So, here you can see that for different ratios of the compounds, it shows different colors. It can be quantitative also. So, here you can see that you have an aptamer and the salt, and then you start increasing the concentration of the salt, and you see 0.5 nanogram to 250 micrograms, and it shows a red shift here, and that is how you see it in the color. So, using these techniques, it is very useful, because you can have a strip which is where you put the analyte and it shows a change in color, you will immediately know that there is this particular compound is present in the analyte matrix. Cavity ring down absorption spectroscopy sensors. In this kind of sensing, what you have is that you have a cavity which has the analyte inside the cavity, and when a laser pulse is incident in it, it gets adsorbed, and after back and forth reflections you have transmitted train of pulses. For uniform absorbers, the peaks decay exponentially. So, for example, tau is tau is the time of flight for this, 1 upon c, 1 minus r, y is the mirror loss, and alpha into L, alpha is the absorbance. So, if you have intensity I0 incident and you get I, you have this expression to solve for, and say for example, for a cavity length of 1 millimeter and the 100 ppm concentration, the round trip precision translates to a minimum detectable absorbance of 10 to power minus 6 centimeter inverse. So, you get this absorption value from the transmission curve, and it is a function of the decay time in the cavity, which can be with or without the analyte. So, this is called cavity ring down absorption spectroscopy sensors. So, let us summarize the talk. Today we discussed what are optimum nanoplasmonic structures for highest enhanced EM fields, and we discussed that it is not the nanostructure where you have maximum enhancement, but in the gap between these two nanostructures, and when you increase the gap basically the coupling decreases and then you have decrease in the enhancement factor. So, while designing a nanoplasmonic structure based sensor, you have to consider this optimization when you make a sensor. We also discussed what is fluorescence and how it can be used for sensing, and then we again utilized plasmonic enhancement for surface enhanced fluorescence and surface enhanced IR absorption. We also discussed what is ESP-LSP coupling, and where we showed that when you have an optimum configuration in the hot spots you can have enhancements up to 10 to power 5 times. That is the electromagnetic field. If you say Raman enhancement, that will be about 10 to power 10 times or so. We also discussed what is a colorimetric sensor. It is very useful, it can be very cheap also, because you do not need sophisticated instruments to go and perform this kind of sensing in anywhere. Say for example, you are in the market and you want to check something, if it is a colorimetric sensor. For example, if you want to check the quality of milk you are going to purchase, what you do is that you have a color sensor, you dip in, you see okay, if this is the color then the milk is not good, if this is the color milk is good. So, it is very easy, but there are certain limitations to it. And then we discussed something which is cavity ring down absorption spectroscopy sensor. Thank you.